Good morning or good afternoon, everyone, wherever you are. Um, for us, it's an important day. So we every year we release data from the OpenScope project, and we had such an event last year, and we're doing this again this year. So I'm gonna present. My name is Jerome Lecoq. I'm an investigator at the Allen Institute for Neural Dynamics. And today we're going to have a second data release under NIH funding. But first, before we dive into this, I want to make sure that you understand that this project is a really teamwork, and it involves a group of people at the Allen Institute as well as across our community. And this is very important to us because we are building this observatory as a community resource. So first, you can see on the top left that the pipeline that is collecting the data that we are going to release today is maintained and was built by scientists, research associates that are collecting the data, managers that are helping make this process smooth and efficient, as well as engineers that built the rigs and software engineers that are maintaining the software that allow for this work to work well, as well as a really important team that are taking care of our mice. In addition, a number of leaders in organizations have led the development of the pipeline, and you see these names on those slides. But there are many that I could not be listed here. And so I just want to make sure you understand this is teamwork, and I'm only one a representative of this group. In addition, the OpenScope project is led by a steering committee. So you see those people in the middle, and we are incredibly thankful for their guidance. We meet with them multiple times a year, and the project that you're going to see today were selected by the steering committee as well as really important reviewers across the community that help guide a selection process. In addition, we also funded by a number of organizations. So first, the Brain Initiative took a leap of faith and funded this platform. I'm incredibly thankful for their support, as well as the Falconwood Foundation that really led the initial first round, and the Allen Institute itself, which supported this work. So take this first home. This is really teamwork. And so we are incredibly thankful for the community support for this project. Okay, so what is an observatory? When we started the program at the Allen Institute, there was a clear vision set by Claire Reed and Christoph Koch of building Observatory of the Mind. And the idea behind this platform is that there's a tremendous amount of complexity in our brain and in animals' model brains. And so to tackle this challenge, we need to collect data set of multiple scales and multiple integration points, be it from the individual diversity of cell types or their activity in vivo. And to achieve that goal, we believe that building observatory of the mind was a key tool that will unlock our understanding of brain. And so we set ourselves this goal of building pipelines that will allow us to collect at high scales and across many animals data sets that help the community move forward. And so that's what we set ourselves to do. And over the last decade, we release a number of data sets on a regular cadence. And some of these are showcased here, but there are many more. So these are two publications on the left. You see a platform that was built for doing large-scale two-photon calcium imaging. This was, I believe, the first large-scale imaging platform for in vivo physiology. And you can see on this figure that the number of animals involved was an order or 2 orders of magnitude larger than a typical study. And so we released data set that were designed at the Allen Institute And this publication you see here is what we call a platform paper, where we highlighted the design of this data set as well as how to access those. On the right, you see the same effort for neuropixel recordings. So we position 6 probes into the visual system of the mouse and recorded spikes across a number of distributed areas, and similarly released data set across hundreds of mice. And those data set had a huge impact. And so you see on the left, this is what we consider the equivalent, a platform in astronomy. So this is a paper that was released by the Keck Observatory a few decades ago, where they show the impact of their platform on the number of publications over the years. And when we started this effort, we were sort of looking forward to see whether the community would take this data set and make new discoveries or push the field forward. And so this is what happened. On the right, this is a recent review put together by Saskia de Vries, Josh Siegel, and Christoph Koch in Eli, where they summarize the impact of those open data set onto the community. And so you see we're sort of enabling a lot of work across the community. But those experiments were designed at the Allen Institute, and we were not operating as a true observatory in the sense of what astronomy is doing. We were designing the project internally for greater use, but we felt that the next logical step was to establish a true observatory, a place where projects are coming from the rest of the community and executed for the greater goods of the community. And so that's what OpenScope is. We consider this as the first observatory on neurophysiology in neuroscience, and we operate by taking projects from worldwide scientists. They can run their own experiments on our platform. So this is either a 2-photon or NeuroPixel experimental platform. And our long-term goals are to maximize the impact of our effort, the platform that we have built, as well as continue to build this community and develop that approach in system neuroscience. So today, we are going to see data release that is coming from this effort. So the component of these resources are listed here on the slides. So first, we give access to the Brain Observatory and data collection pipelines. That means that we execute the experiment, we execute the mouse work, and we release the data to teams that submit projects. These projects are submitted to a review process on a yearly basis. It's almost like as a grant. You're getting the teams that were selected based on the merit of the scientific project. The review process was blinded, so it's really science that matter. And we execute this experiment for free. And similarly has how other observatory operate in astronomy. In addition, we built an integrated cloud and analysis ecosystem in partnership with other projects supported by the NIH, including the NWB format and the DANDI platform. So the data that we're releasing today is available on DANDI. In addition to establishing this observatory platform, we operate as a technical hub. That means that we organize this event today where we disseminate the science of those projects, as well as welcome scientists from all over the world comes once a year to a technical workshop. I'm going to talk a little bit about this later. So the intent, our intent, is to develop a community around the platform to help neuroscience integrate and build shared projects. So this is the technical workshop that I alluded to. This year is going to happen in July between 9 and 11. It's a 3-day workshop where we show everything from the pipeline that you see in the middle with real how we actually operate animal surgeries with the implant. You see a picture on the left, and 40 people come from all over the world. We had several of these events. They're really great fun. And so this year is going to be in July. I think we are currently selecting the the 40 people that will come to Seattle. But the application window is closed at this point of this year. Okay, so today is an important day for us. So we are disseminating the data that we selected more than a year ago. And our intent is to facilitate data reuse. So we want to, for the community to integrate better one another, for people to work well together. And it's important that we help define the rules of that data reuse. And it's up to us how we do this. And I personally hope that we foster collaborations versus competition. I think the teams that you're going to see today really care about their project. They put a lot of thoughts and effort into designing those experiments, and they continue to do data analysis on those assets. And so what we hope that this data release process achieve is bring together people. And so if you are interested in this project, I really hope that you reach out to the team. And so we're going to facilitate the process as much as we can. So there's a Slack channel here where you can join and talk to all of the OpenScope community. And so if you have some idea of analysis that can be done on those data that we released today, I really hope that you try to reach out to the team and build analysis together instead of competing with one another. I think it will be healthier, and I think everybody will be benefiting from building that community. So our schedule today start with Pamela Renago. So she's going to talk to you about the temporal barcode project, that's how we call it. And the full data release happened, I think, yesterday evening. So we released the embargo on this data set. Michael Berry was scheduled to talk after Pamela on the temporal sequence learning project. Unfortunately, he had a last-minute schedule problems, and so we had to cancel that talk. We are going to work with him to record this video and append it to the webinar. So we'll go directly with the OpenScope Depot team. So I apologize, some names are missing here. So Chinmay Puradare, Mayank Mehta, and Siddharth will talk about the Vision to Hippocampus project. And these experiments will be released in March 26, 2025, so in a few weeks. Our policy is that we release dataset 1 year after the team had access to the full dataset. And so for this team, it's going to be on at the end of March. But we'll still present it today along with the Databook that Carter will talk about. So this will be the last part of the presentation, where we'll go over some of the tools that we put together to facilitate data reuse, including some Python notebooks. And I'll finish with some last exciting development around predictive processing and the community project that we are building in 2026. Okay, so that being said, I'm going to give the floor to Pamela Renago.
Okay, you have to unshare for me to share.
Thank you, Pamela.
Okay, are you seeing my slide?
Yes.
Okay, great. So I'm very excited to be here today, and very honored that the Allen Institute entrusted so many resources into an experiment that I proposed. And my goal today is really to share with you how the experiment was designed and why, and what kind of data are in this data set, because the motivation behind this project, as I'll unfold, is really to fill what I saw as a gap in the Allen Institute open data set ecosystem, and really in the world open data ecosystem, which was a dearth of sort of comprehensive brain-wide recordings of responses to visual white noise stimuli. And so Jerome already introduced the very large team at Allen Institute that supports this project, who I have not tried to list here. But I did want to highlight a couple of individuals who very particularly helped with this project in my lab. Let me turn on my laser pointer. Right. So in my own lab, DNVS is an undergraduate at UCSD, worked with me early in the project to analyze the pilot data set and refine the experimental design. And Sid is in my lab now, is helped us develop the Databook that Carter will be showing you later, and is working on the barcode analysis that I'll describe today. And at the Allen Institute, I had the pleasure to meet people on the entire technical pipeline and see their amazing expertise and rigor of how they each do their job. But I've really interacted quite a lot with Jerome and Severine and Carter. So a special shout out to them. Okay, so without further ado, at least at the time of my entry into the field of neuroscience, most of how we thought about neural coding and vision and in really neural function in many neural systems was in terms of the firing rates of neurons, because it's quite obvious that different stimuli or different conditions modulate the average rate of firing of neurons. And this is the level of analysis that allows us to derive things like tuning curves and receptive fields, and is definitely a major part of the story of how the brain works. But in the late 20th century, people started using white noise as a probe stimulus for the nervous system more often, because it has very desirable statistical properties, and it's a maximum entropy stimulus. It's a completely unbiased stimulus. And so this is an example of recordings from the H1 neuron of the blowfly from de Ruyter van Stevenink and the Bialik, showing that if they showed white noise motion stimulus, so moving stimulus that changed to direction randomly at every time step, this neuron will fire spikes at some time in every trial. And if you keep showing new white noise stimuli in every trial, you get spikes at different times. And if you average the firing rate as a function of time over all of those trials to get a peri-stimulus time histogram, it looks pretty flat. The cell is firing like 50 spikes per second to this kind of stimulus. But if instead of showing a new white noise stimulus every time, you show the same white noise stimulus over and over, they revealed that the neurons are firing those spikes at incredibly reproducible times in every repeat of that very random stimulus input. And another way of looking at that is that the PSTH has very clear peaks and valleys. So it has this somewhat sparse representation where the timing of the spikes is clearly something that's being reliably controlled by the stimuli. Similarly, around that time, Michael Berry and Marcus Meister published this paper showing very similar kinds of responses to white noise stimuli in the retina of the salamander. Barakas et al. In Tom Albright's group showed similar responses to randomly reversing motion stimuli in the area MT in the monkey cortex. And we did a very similar experiment when I was a postdoc in Clay Reed's lab, showing full field flicker white noise, either with unique white noise sequences or repeated white noise sequences in the LGN of anesthetized cats. And in all these cases, what you see when you show the same white noise over and over again is this pattern in the rasters that just looks literally like a product barcode that you see on your loaf of bread or toothpaste in the store. And that's why we've dubbed this the temporal barcode project, completely unrelated to genetic barcoding, which is completely unrelated use of that term. But I think you can see why we think of these as barcodes. It's another way of saying that the spikes are fired at very particular times and not others, or the PSTH, has very discrete peaks. And it's not just a novelty of responding to a sort of, in all of those cases, I should say, the noise was sort of one-dimensional white noise, which means there was just one stimulus parameter that was varied randomly in time. But responses like this can occur in more naturalistic conditions. So these are just unpublished data that I collected from the CAT LGN, where this red square shows the cell's receptive field, and either spatiotemporal white noise flicker or just playing natural movies also evokes these barcode-like responses, at least in some times during natural movie presentation. So this is something that really occurs during vision. But even if it didn't occur during vision, it revealed something that when I first saw it, I was very astonished by, which is that not only can we get this very precise and reliable response from many repeats of the same stimulus to a given neuron, but we sometimes will see the almost identical barcode in the response of another neuron. So each one of these groups of rasters, so like one group of rasters at top is from one neuron, and then each of those groups is a different neuron. And I've color-coded them because all of the ones that are drawn in red have at least certain bars in the code that are shared in common much more than is expected on chance, whereas the blue ones, they don't share that statistical matching with the blue ones, but the blue ones do with each other. So when we saw this, again, this was the work I did in Clay Reed's lab over 2 decades ago, it suggested to us the idea that maybe these barcodes, even if they're not used to transmit visual information in real life, although I think they may be, but even if they weren't, they're obviously a reproducible thing that development and evolution has conspired that some cells have this particular code. And so maybe it could be taken as just a fingerprint of neural biophysics, that these neurons have particular time constants and integration time windows and intrinsic properties that cause them to fire at these specific times. And there's been some selective pressure for that configuration, that biophysical configuration, to be repeatedly achieved. This is just a close-up example showing that 2 neurons that were recorded in 2 different cats with really nearly identical barcodes. And this is one from Berry, Worland, and Meister showing that they found nearly identical barcodes or PSTH peaks in responses to the same white noise stimulus in 2 different vertebrate species. So this idea that these might be markers for cell types came to our mind. And so we had this idea that we proposed that we could take the barcodes of 2 different neurons and quantify how similar they were using something like an edit distance metric. And this is a figure taken from Victor Purpura. And this is just a way of defining the similarity between any 2 spike trains by the minimum number of steps it would take to convert one into the other, with certain conversion rules. And I won't go into the rules of this particular scheme, but if you can represent the distance between any 2 examples in a metric space, then you can hierarchically cluster, or you can cluster using a hierarchical method and determine whether there are, as this cluster indicates, discrete groups that are clearly, quite separate from other groups, and look at the sort of space of neural codes. So we proposed this project using the passive viewing NeuroPixels platform. The idea was to record spikes from as many neurons as we could in as many visual areas as possible while showing the same white noise stimulus repeatedly. We were very interested in recording in the subcortical areas, and especially the parallel pathway of the visual system that goes through the superior colliculus, which has been much less studied in the white noise paradigm compared to the LGN to V1 pathway. Our stimulus is one-dimensional white noise, which means we just have one. It's not like a checkerboard that people use to map receptive fields. It's just one stimulus parameter varying in time. And that's shown either as a uniform full field flicker, which was used in many of those earlier experiments I just showed you, which is the whole field is one uniform luminance, and the luminance changes from frame to frame. We were worried that the higher visual neurons wouldn't respond to uniform full field, so we also did phase reversing standing grading. So there was a spatial pattern that didn't move, but the contrast of that pattern changed in time with the white noise sequence. And because we can optotag, we also did, and we used the 2 available lines, SST and PV. So all of the mice in this project also have one of those 2 cell types optotagged. And so this is just the overall blocking of the experiment. We have neuropixels electrophysiology, but half of the mice with the standard configuration that hits all of the cortical visual areas, and the other half with a novel configuration that was developed, collaboratively for this project that really prioritized LGN and superior colliculus. We start with full field flickering white noise, which is what I'm going to talk the most about today. And then we have standing grading, whose contrast is reversing according to the same white noise sequence that we use for full fields. And we did that same white noise sequence for many different orientations, phases, and spatial frequencies of gratings. And so that ends up taking the bulk of the time in the experiment. And at the end, we flash Gabor's the standard receptive field mapping stimulus. And at the end, there's opto tagging. So that makes up the roughly 2 hours of recording time in each mouse's data set. So for the repeated stimuli, in order to have enough repeats for the kinds of analyses people might want to do, we limited it to an 8-second duration. And half of that time, we had a binomial white noise. So in every video frame, it was either all on or fully off at chance. So this is a short segment of binomial white noise. So the distribution of values is just -1 and 1. And the second half was drawn from an exponential distribution. And the reason we chose this is because it closely mimics the distribution that we found in a natural scenes analysis that I won't get into. I thought it would be better to have a well-defined statistical distribution than to use the natural scenes. And so that's what we used. The monitor that's used in these experiments is 60 frames per second, but we opted for a 30 frame per second update of the stimulus. And there's always a trade-off. If you update too frequently, then the stimulus doesn't drive the cells as well. If you don't update frequently enough, you might not get as sharp, precise responses as we might have. So that was the compromise that we picked. And then, so that's the most of the experiment. There is also a very small amount of time devoted to unique segments. There are some analyses like spike triggered average, where one might like to have a much longer representation of white noise, but you don't need to repeat it. And so we have 120 seconds, what we call the unique full field, half of which is binomial and half of which is a Gaussian distributed. And the Gaussian white noise we chose from this paper from the Gullish lab, who had already made their data open, because they have used this exact stimulus for studies of the mouse retina in addition. So it will allow us to compare our subcortical responses to their retinal responses. Okay, so what did we find? We found that in the awake passively viewing mouse preparation, that neurons still fire barcodes. So it isn't just something found in retinas in a dish or anesthetized LGN. And these are just a few units. This is one from the lateral geniculate nucleus. This is one from the pulvinar. And this is one from primary visual cortex in layer 5. And we actually found barcodes in every visual area, and some visual areas and layers, most of the cells had some kind of barcode. Another way to say the same thing is that if you took PSDHs of these neurons, they have very sharp peaks. They're relatively sparse, and we can use statistical criteria to define which of these peaks is significantly above chance, and we'll define the times of those peaks as barcodes. And our Databook that you'll hear about later goes through a sort of a simplified analysis for how you might pull a barcode out from these data. So our preliminary observations from the data set so far is we did replicate the result from anesthetized animals, that we have these barcodes or sparse temporally precise responses in the LGN, but we also found them in the colliculus and LP. And in every visual cortical area that was recorded, and in every layer of visual cortex. And for some visual cortical areas, most of the units have a barcode. We also saw what looked like barcodes in some areas that we didn't think of as visual areas at all, but I'm not going to list them now because we'd still like to really nail down whether we're totally sure that they were in the area we thought they were. The other huge surprise for me was that most and possibly all of the cortical neurons that had a well-defined barcode with flickering gratings also had a barcode for full field flicker. So it turned out that full field flicker was perfectly fine for driving cortical neurons. And that's why we've been focusing this presentation and our early analysis on the full field flicker. So the benefit of using full field flicker is that every neuron that we record within one animal and across animals, no matter where their receptive fields are, all experience the identical visual input. And so that we can compare their responses without having to take into account possible differences in their visual input. And we also find that in some metric spaces, at least, you can use hierarchical clustering and see some evidence of hierarchical structure in the clustering of these neural codes into classes. So what we're working on going forward, there's a bunch of issues, and I'm listing these partly because we would love to have more collaboration. So spike sorting is, of course, a hard problem, and these are neuropixels 1 recordings, and the interpretation of analyses like this can be affected by how confident we are whether these neurons are really perfectly well isolated. So I think there could be advances in the world of spike sorting could really support stronger conclusions in this project. Like all neuropixels recordings, we have some instability of recordings where neurons drift off the cluster from the beginning to the end of recording. So we see changes, from beginning to end within a neuron, and it's difficult to distinguish those from true biological non-stationarity of the unit's responses, but both of those are things we need to really think about and address. If we're going to talk about a neuron having a barcode, what is it that we can extract in spite of those sources of variability? There are many different possible similarity metrics, and it's not yet clear which one really captures the way in which related barcodes are related. And then there's many different ways of clustering. And then there's the big can of worms of statistics of making sure that we have really robust and meaningful null hypotheses to understand which of our results will be expected on chance with more realistic than trivial null hypothesis. So, but part of why we proposed this and the argument we made in our proposal for why we thought this should be done was not just for that one experiment. We don't know whether there's going to be clustering of cell types. We didn't even know if Cortex was going to respond. So that part was sort of a high risk project, but these kind of white noise data are useful for so many other kinds of analyses that could not previously be done with any of the other Allen Institute data sets, like spike-triggered average, spike-triggered covariance type analysis, calculations of entropy and mutual information by many different methods, sparseness analysis. Obviously, you can do studies of reliability or Fano factor in many other kinds of stimuli as well, but there are things to be learned from the white noise stimuli that I think will be unique. Talking about measurements of spike timing precision, spike count reliability, I guess I put twice. Generative models to try to predict when every spike will happen based on the input stimulus and a model of the cell. And I'm sure there's many more. And so we thought that the data set could be broadly useful by many groups for many kinds of analyses. And that was really the motivation for the project. And so I just wanted to end by saying that we would be thrilled to have more collaborators. Just recently, Tatiana Sharpie and Ryan Roenkamp at the Salk have started working on clustering analysis with us. But anybody who's interested in digging into these data, we'd love to hear from you. And I'm not sure how I am on time, but I'll stop there and take any questions. Jerome, you're muted.
I don't think we have questions.
Okay.
Are there particular brain areas you're particularly interested in this? Because I know we recorded the supracorticulus, but do you think that these barcode could be helpful to understand visual flow across those areas? Or
happy to expand on that. Yeah. So one of the things that's exciting about recording from all of the visual areas and both of the main visual streams is that we can ask questions about whether there are channels of information flow that are, for example, particular barcodes or related sets of barcodes that flow from one area to the next in the visual hierarchy, or that are unique to one or the other visual stream. And also just whether the things we've come to understand about the role of spike timing in coding in the context of the thalamocortical pathway, do they even apply, or is it a very different story in the colliculo-pulvinar pathway? So for our preliminary results are that the things that we learned in the thalamus and cortex do also seem to be found in the colliculus and pulvinar, but differences between them, remain to be teased apart. And for me, this is a long time and really major interest of my lab. What is the implication for mammals that we have two separate visual streams that are each capable of controlling our visual behavior, and that I hypothesize, subsume different essential functions for animal behavior.
We have a couple questions in the Q&A. What sort of challenges are you facing with the new models?
With the what?
With the null model.
Oh, well, so it's we're just trying to be very careful and still looking for not satisfied that we're done constructing the question of how often would we expect to find a certain amount of relatedness by chance or a certain amount of clustering by chance to adequately take into account all the statistical correlation structure in the data. So what do you expect on chance, given that there are correlations between neurons, that there's non-stationarities, and that those non-stationarities can be correlated, and just making sure that all of those have been adequately built into, I don't really see any hope other than a bootstrap kind of approach, but making sure that we've adequately built those in, because it's just incredibly easy to fool yourself about it's an exploratory study, I should say. So we're not testing really a hypothesis so much as describing the data. So there is definitely a massive multiple comparison issue or data dredging, thing. And we just want to make sure that we are careful and that we don't fool ourselves into seeing patterns where there are none. So
I think the next question is related, so you might be able to answer it directly. Aside from non-stationarity, is there also heteroscedasticity or other issues?
We have not dug into analyzing that yet. So there certainly could be. And like I said, right now, one of the reasons that's hard to even get started on is because where we see things, it's hard to tease apart what's a technical limitation in our recordings of the neurons and what's a property of the neuron themselves. So
I think the next question I might be able to answer, but you can wait on it. Are the electrode tracks reconstructed and units mapped on the anatomy for the recording, or is this still a work in progress?
No, this is part of the amazing work that the OpenScope team contributes to this work. So they have mapped all of these neurons onto the common framework. They've mapped them. We didn't have enough time in the experiment to include the stimuli for current source density, so we have not done that, but they've been tentatively assigned to layers just based on depth, and assigned to visual areas based on the common framework already. And a spike sorting has been done with a whole bunch of validation criteria provided already.
There's a question from Chinmay, and great proxy for the next talk. Does your data set have hippocampal cells recorded on the probe that went to LP, for example?
Yes, so anything that the electrodes passed through, we've recorded and clustered and analyzed, and we're putting them all into the analysis. And I have seen cells that were assigned to the hippocampus that did show barcode responses. But like I said, those cases I haven't gone back to look at the tracks to see if there was any ambiguity about their localization. But yes, there are, like the other neuropixels data sets, there's a lot of hippocampal neurons because the electrodes go through hippocampus.
Okay. Don't think I see more questions. Well, thank you, Pamela. It was just such a fun collaboration, and I hope everybody please reach out to her if you want to collaborate or are interested. This is really important. And so we're going to proceed to the next talk. So the next talk is going to be presented by Chinmay, Mayank, and Siddharth. I think it's from UCLA and now UCSF. So I'm going to let you start.
All right. Can you hear me?
Yes.
Great. So thank you for picking our project. We are enormously grateful to Allen Institute. And thank you too for putting up us, dragging you into hippocampus and dragging us into visual cortex. So this is somewhat unusual for Allen. So let me give you a broader background. As you have seen that since the discovery of Hubel and Wiesel, we look at visual cortical neuron responses to specific stimuli. And since then, there's this huge circuit that has been built up where neural responses evolve in interesting ways through the whole circuit. But what is not commonly appreciated is this, that at the top of the circuit is the hippocampus and entorhinal cortex. And people don't usually study them in the context of representing specific visual stimuli. What they do study is, since the pioneering discovery of Brenda Milner, who is still going strong, is that hippocampus is required for episodic learning and memory. It's hard to describe what is an episode. We know what is roughly memory, but what is an episode? That we need to understand. And the race was on, and then people found actually hippocampus contains a cognitive map or a GPS system of the brain. And in particular, it's believed that the hippocampal places represent allocentric, abstract location and not sensory stimuli received at that location. So something happened between visual cortex, which is in the egocentric coordinate frame. The animal receives it at a certain place, even retinotopic, and all of a sudden it went somewhere else. The story got summarized very briefly that in mice, there are abstract location of place. This is because there is no direction selectivity. The neurons don't seem to care for what stimulus the mouse is looking at. Hence, these are not sensory responses. These are supposed to mediate navigation. And all of that is mediated by an 8 Hertz theta rhythm, which is crucial for everything, including neural coding and plasticity. It turns out that this is measured in classic tests like water maze or the standard maze that the rats run around, where we say it's very hard to measure what are the stimuli. Whereas in primates, freely behaving primates and humans, these experiments are extremely difficult. Position selectivity is weird, found in 1D, but not 2D. These neurons, all of them, whenever they are selective for direction. They are strong sensory evoke responses. Many of you have heard of so-called Jennifer Aniston cells, which are actually found in hippocampal area CA1 in humans. This is supposed to mediate episodic memory, and the theta rhythm is quite weak, about 4 Hertz. And the classic test of function in hippocampus in humans is positive memory, such as what did you have for lunch? So 6 out of 6 things don't match between rodents and primates and humans, which is not true for visual cortex at all. So how come? So we hypothesize that the reason might be that when we think where you are, this is a top-down view of a maze. When you think where you are, you localize yourself with respect to the views around the walls in the room or the environment or distal visual cues. But rats have a very strong sense of smell. They smell in stereo. They can be smelling all things around them. They could be paying attention to tiniest sounds. They could have vestibular inputs. And the most important, when the rats are running, their nose is next to the ground and they are picking up tiniest of stimuli on the ground. So unlike us, mice receive very strong olfactory and barrel cortical stimuli. We don't even have the barrel cortex. So can we eliminate these to find a comparison and understand what is an episode, what is a space, and how do you go from vision to hippocampus? So we started this project roughly 12 years ago, where we built this unique non-invasive virtual reality system where the rat runs and he can see the stimuli all around him, but because he's in VR, all the other smells and textures are uncorrelated with vision. They are still present, but they don't have anything to do with what the mouse is doing. We can do many, many trials. We can do very reliable electrophysiology while they are doing this. And most important, we can now study rodents, primates, and humans in the same apparatus, because the rat can be removed and the same apparatus can go around a human head or a primate head. We published a fair number of papers, which are all available on Lab's website. And very briefly, I want to show you how we went towards vision by using this. The first surprise that we reported roughly 10 years ago, that when the mouse, or in case of a Longevin's rat, ran around in the real world, there were nice place cells. This blue line is rat trajectory. These dots are spikes. You find very nice place cells when the rat runs in real world, like everybody shows. But the idea was that place cells are path integration or vision and locomotion combined in virtual reality. When the rat runs using vision and locomotion, which are intact, no place cells. In contrast, when the rat ran in virtual reality, though there was no spatial selectivity in space, there was a very strong direction selectivity towards a specific visual stimulus that we could control, including the width of the tuning curve. So at that point, we thought, let's go back and get rid of virtual reality entirely. So we sat down a rat and we simply took a bar of light and moved all around this rat. Just a bar of light going around, no rewards, no task. We gave him random rewards so that he remains somewhat in a theta state, as they call. What do the neurons do? And Chinmay, and we found that when the rat is sitting here, the bar of light is going around. 0 degrees is the stimulus in front. He can see all this region, these areas he cannot see that behind him. And we moved the bar of light either clockwise or counterclockwise, just in the same direction. And we found actually neurons. These are all place cells because when we took out the rat from the apparatus, let him run in the maze, they all showed spatial selectivity. So this neuron is tuned when the bar of light in the counterclockwise direction fires to the right of the rat. This neuron fires slightly to the left. This neuron fires again to the right. And there are clockwise tuned neurons as well. These neurons don't have selectivity in the other movement direction. This neuron in the clockwise direction fires to his left. This fires to his right. And this neuron fires maximally when the stimulus is back. These neurons show cortical magnification-like properties. More neurons are tuned towards the front than in the sides or back. The tuning curves get wider. So this started to tell us, hey, this is looking like a very specific stimulus response, not too different from visual areas. So then our first collaboration with Allen Institute started with the previously recorded data from Allen Institute. They had a black and white movie that they had shown to rats where they were just sitting there. The movie is 30-second long, publicly available data. Chinmay and I dug into this. And here is one neuron from CA1, but this selectivity is found all over. So what you'll see at the top is the raster plot of one neuron for 900 frames. The movie was going at 30 frames per second. And at the bottom, you'll see PSTH. And what we saw in this mouse, which is head fixed, monocular black and white movie, no task, no rewards. You see this very funny activity pattern where you might see something that might look like barcodes to Pam, perhaps. I don't know if that's sharp. And then there are solid periods for which the neuron is active very strongly. And then there are periods in which it completely shuts down. And these responses we feel are episodic because if you take the same movie and scramble the images and repeat it, they're gone in hippocampus, but the responses are present in visual cortex. So interesting, lots of papers out of this. So I can briefly summarize this and see where we are going after this. So these were supposed to be abstract allocentric plates from 1971. When we put them in virtual reality and they walked on a linear path, we got both place and direction cells like they see in primates. If they walked randomly, they only encoded delta t or time. I didn't go into the details because this is old stuff. All 40% of them show direction selectivity when the rat's navigating in the virtual water maze task. For a positive reward, these responses are related to behavioral decision. Even theta rhythm changes becomes 4 hertz. So these results are unpublished, so I will skip that. And then the waves, which is just no virtual reality, rat just sits there, the stimulus goes around, and you see selectivity to a single stimulus as well as stuff going around. So these things are not looking abstract. There are specific responses to vision and locomotion and nothing else right now. We haven't looked too much about reward yet, but that's somewhere else. So to summarize, in the real world, rodent neurons have abstract position, no direction selectivity, not sensory response, and the test is a negative memory test called Morris-Water maze. In primates and humans, none of them matches. But when we test rats in this case, using only vision and no negative rewards, everything matches. You get position selectivity in virtual reality in 1D, but not 2D. Strong direction selectivity, strong sensory evoke responses, even just a moving bar of light, and episodic responses. This is practical purposes, which we are following up. The reason Alzheimer's therapies may not translate from mice to humans may be because we are testing different kinds of mazes. The virtual reality with vision alone may be a right test. And the boosted theta rhythm might be useful, but that's a separate story. May be useful as a therapeutic. These are the papers that we have published. All of them are available on the lab's website. So at that point, we thought that, we need a bigger boat or we need people with a bigger boat. So we were very happy that Alan folks collaborated with us and took on our questions. So what are the questions that we are going after? First question is there this vectorial selectivity to bar of light? Where did it come from? First of all, to do that, we want to check if it exists in head-fixed mice with a screen only on one side of the head, not everywhere else. So the kind of space and so on is kind of broken. Mouse never did a virtual reality, so there's not even an imagination of some space or something. Where do they come from? Are they in CA1, if they are found? Are they in dentate gyrus, which is the input to hippocampus, or in CA3? Moving further up, what about entorhinal cortex, where there are all kinds of interesting things, is it like grid cells? Do the responses exist in the entorhinal cortex as well or not? Then we want to do two things. One is to go directly towards simple sensory stimuli like a bar of light. We use various stimuli, but this was the most common stimulus we used. And more complex stimuli, which form a natural stimulus / episode. There is a chance that the mouse may have some idea about that. So with that, I will stop sharing the screen and let Siddharth take over the next part.
So I'll describe the experimental design briefly and some tuning curves in CA1 and CA3. So Allen Institute has this amazing setup in which they have a mouse running on a disk, and the mouse is seeing a screen but monocularly. So it's not seeing, so the screen is not surrounding the mouse. And one can actually record the running speed. One can actually find the pupil diameter. So these are the two major behavioral cues that we record. And this is how a schematic looks like. So there is a running disk for a head-fixed mice, for our head-fixed mice, and there is a screen on which we present different kinds of stimuli. The OpenScope Databook has description of all kinds of stimuli. I will give description of two qualitatively different kinds of stimuli that we show. But before that, in each of the experiments, there are either 3, 4, or 5 probes. Mainly, we have targeted hippocampus. So the pie chart shows a typical distribution of the top 10 regions that we record. We have recorded a ton of CA1 cells, a ton of entorhinal cells, and a ton of cells in visual cortex because the neuropixels are going down. And this is how a typical configuration of our probe looks like. So this is an example of a simple stimulus. We mean by a simple stimulus is the stimulus we have looked at before in rats. This is a moving bar of light. It moves from left to right in roughly 4 seconds. It has roughly 120 frames. And we look at the tuning curves of neurons deep in the hippocampus for these cells, and we indeed find tuning curves which look pretty sharp. And actually, Chinmay will talk about how we classify whether a cell is tuned or not using bootstrap method. But we find tuned cells in CA1 and CA3, which confirms our earlier result, which Chinmay had seen in his Nature paper. Even though the visual stimulus is presented monocularly and the animal is head fixed, still in mice, we do see tuned cells in both CA1 and CA3. We also present more natural stimulus, which resemble what mouse will see in real world. For example, we have stimulus like eagle swooping, we have stimulus like other kinds of movies. And in this case as well, we do see tuned cells, both in CA1 and CA3. Although the tuning curves are pretty complicated, but they are indeed tuned. And now I will hand over to Chinmay to describe the next part of the story.
You're muted, Chinmay.
Sorry, can you hear me now? Am I audible now?
Yes.
All right,
Yes,
perfect.
you're audible.
Sorry, yes. So once we saw selective responses in CA1, we started to dig into the circuit and go one step upstream of that. So the entorhinal cortex is the gateway from cortex, in this case, particularly the visual information, to the hippocampus. But as Mayank described previously, entorhinal cortex in rodents is studied in spatial navigation tasks. So this is a typical response, one of the most famous responses of an entorhinal cortical neuron. This is from medial entorhinal cortex. So there's two parts to it. And what we are looking at is the firing response of a cell when the animal was running in a square environment. And red areas are where the neuron fired a lot. Blue areas is where the neuron didn't fire. And this is the famous grid cell. The firing pattern of the neuron is on the lattices of a hexagonal grid, so to say. In the same brain region, in different sublayers, there's these other cells which are head direction cells. So there's not much of a grid pattern. We're still looking at a firing response from the top, but this neuron fires a lot when the mouse is facing to, or the rat is facing towards the left side. So this is what is known as a head direction cell. And then there's other sets of cells which are known as boundary cells. What these cells do is they care for the edge of the environment. And interestingly, if the environment is circular or square, they care more or less for the similar side of the environment. So these are what are known as boundary cells. And the entorhinal cortex, where all of these cells are predominantly studied, is the medial entorhinal cortex. And it's hypothesized that it provides input to the proximal CF1, but that's not the entire story about the entorhinal cortex. There's this lateral entorhinal cortex, which is believed to get input from different parts of cortex, and it feeds into different subregions of CF1, and there's beautifully described circuitry here. So the question I wanted to ask was, do entorhinal cortical cells respond to different positions of the bar of light? So what I'll be describing is, as Siddharth mentioned, a simple visual cue. It goes from the front of the animal to the back of the animal and then comes back. The screen at the rectangle, as we see, is positioned on the right side of the animal. So even if it looks left to right for us, with the warping and the proximity of the screen, it looks like front to back for the mouse. And similar raster plots, as Pam showed and Mayank showed for our previous work, what we see here is a response of a neuron across 60 trials. Each dot is a spike of a neuron. This neuron was recorded in entorhinal cortex, the medial entorhinal cortex, layer 6, and the red trace is its average firing rate response. So it goes from front to back in 2 seconds, comes back from the back to front in another 2 seconds, and this happens trial after trial. You see very nice responses. This cell, for example, cares for somewhere in between, so it probably cares when the stimulus is to the right of the animal. And interestingly, it fires at that location when the stimulus goes from front to back, as well as from the back to front. The interesting part further is that the responses are not exactly the same. They're pretty much at the same location, but when it comes from the back to front, which can be thought of as a temporonasal motion, the firing rate on average is slightly lower. So it cares for the same location, has tuning in both directions, but it's not exactly the same. There's another cell from the layer 2 of entorhinal cortex, and this one also cares for both directions. There's much more difference in the average response of these cells in the two directions. Another cell, one of my favorite ones, it really likes when the white bar of light is behind the animal. It fires a lot when the stimulus is at the back. In later trials, interestingly, it started to fire even at the side of the animal, but only in one direction, not both. And this is 3 more cells from other subregions of the entorhinal cortex. So the Allen Brain people have done an amazing job of recording from multiple parts of the entorhinal cortex, as well as assigning recorded neurons to sublayers and medial versus lateral entorhinal cortex. And we see a wide variety. I'm just showing 6 cells here, but we see a wide variety and rich set of diversity in the tuning curves of these neurons. So we conclude that the entorhinal cortex encodes different positions as well as differs between the movement directions of a passively moving bar of light. To quantify the selectivity briefly, we use a sparsity metric. Rn here is the firing response in the nth bin. So if you quantify actual sparsity for a neuron which is tuned like this, you get a high value, whereas you can bootstrap it. So you misalign different trials and you get a flatter response, and you quantify its sparsity, and you get a sparsity which is pretty low. And you can z-score it to the actual sparsity using the shuffles by performing 100 different bootstraps. And that's how we quantify something known as a z-score of tuning. And what we are seeing is that across brain regions in the entorhinal cortex, across lateral as well as medial entorhinal cortex, there's more than 50% of cells which are tuned for the different positions of the bar of light. And to appreciate how significant it is, the black dotted line here is what would happen by chance, what would happen in the shuffle data. So it's massively different than shuffle. And now we are digging into the other properties of this tuning. So majority of cells in the entorhinal cortex encode different positions of the bar of light. They do so significantly. All right, so we'd like to thank the Allen Brain, as well as our institutes and our funding, specifically the neurosurgery and behavior team at the Allen Institute, Jerome, Severin, Ryan, Hannah, Kyla, and the other RAs who did a great job recording neurons. Time is short, so we can't describe all the cool things that we see, but there's a lot going on, and we invite you to get in touch if you want to collaborate, dig deeper into the data with us. Thank you.
Thank you. I have a quick question. Because you're sort of introducing this network as downstream of the visual processing in the cortex. Do you anticipate these strongly tuned responses to influence the responses in V1, or do you think that happens, or is that too far at this point?
You mean like a feedback from,
Yeah,
say, the hippocampus to the visual cortex? So the Van Essen diagram we like to show because it shows our brain region at the top, but it's only half the story because there's always feedback amongst cortical regions from hippocampus to cortex. So this information should be feeding back to there's pathways in the brain which would feed the information back. If the feedback reaches from hippocampus versus simple visual stimuli, seems unlikely because the visual cortex has stronger responses to this. But it could be interesting to think of that feedback when, say, you have a bar of light and you're missing something. Maybe for a second or two, you stop the stimuli, you hide the stimuli. And one could conjecture that that is when the higher brain regions would feed into the lower brain regions and say, well, don't worry about it. We know that this is what is supposed to happen. The screen might have gone blank for whatever reasons, but they can feed back to the visual cortex or higher visual areas to say, don't worry, this is what the stimulus should have been. They'll fill in the blank. We haven't addressed it, but I wouldn't rule that out.
the predictive processing, essentially, interpretation. We have a question in the Q&A. Concerning the difference between theta in rodents and primate humans, the Frank lab recently found strong correlation of theta peaks in rats and fall touches to the ground, which also conveniently explains the theta rhythm and positive correlation to running speed. Have you looked for this in your dataset? Could you be able to do this with deep lab cut of the video data?
So we actually looked at it quite a bit when we looked at this theta rhythm difference in virtual reality and real world. There are about 20 extended data and then another 30 supplementary figures. So it's buried somewhere there. So it's known actually for quite some time that theta rhythm is quite complex and it comes from many places. One of the places people have suspected is whisking. Other place people have suspected is sniffing. And then other things many people have described recently, such as footsteps, which could be synchronized with whisking and sniffing. We don't know. So the surprising thing actually to us is that the rat's movement are exactly the same as far as we can tell. He's still running in virtual reality at more or less the same speed as in the real world we measured it. We measure their the rewards, the length, the duration for which they run is also the same. And still, theta rhythm changes by twofold. Goes for 8 Hz theta rhythm is still present in virtual reality, but but the additional 4 Hz rhythm appears, and it appears only on a subset of electrodes. These other rhythms, for example, there is a rhythm called heart rate related theta rhythm. And heart rate related theta rhythm appears actually in some different part of hippocampus, not in CA1. So it's a massive literature, and theta is influenced basically by a lot. I could say the rat should be called theta rather than a rat, but separate story. It appears even during REM sleep when there is no footstep or any such thing.
Okay.
So yes, we have the video in this experiment where the footsteps of the mouse are captured and it's synced to the data set. So one could do deep lab cut to assign so everything is there in this particular data set. There's footsteps which are tracked. There's a side view, so you can track the front paw and the back paw for the left side. You can see the back paw of the right side, but it will get occluded by the mouse because it's a side camera. There's LFP and there's spikes, and everything is synced together. So yes, one could get into footstep rhythm in this data set.
Potential collaboration.
Yeah.
Next question. Have you considered doing loom stimuli? 1/3 of retinal ganglion cells in mice seem to have this tuning. So many of these go to subcortical region aside from LGN. The tuning from the bar that is behind the mouse makes me curious if that will be there.
Yes, so there is a looming stimuli in this data set. There's a white disk which expands and contracts. Now, Jerome, you could correct me if we are wrong, but my understanding is that the bar of light goes from the front to the back, but even the back is part of the receptive field of the right eye, like it's centered on the mouse's right eye. So my
That's probably
understanding
correct,
was
yeah.
there's very little positions, or there's basically no part of the screen, and hence there's no part where the bar of light goes, which is completely invisible to the mouse in this experiment. But when we did it in rats in the VR arena, there was definitely like 90 degrees at the back where the bar of light went, and the neurons were still having peaks there. Now we don't know if it's a response of, hey, the bar of light vanished and the neurons are responding to the vanishing of the visual cue, or it's like if it's a prediction or expectation that there should be still the bar of light at the back, or is it actually something else? That part we don't know.
Would you consider the eagle stimulus or looming stimulus?
The looming stimulus, as you mean like a threat?
Well, because I recollect there's a portion of the video where an eagle is coming down.
Yeah.
So that's very analog to a looming stimulus.
Yes.
Would you say?
I think that's why we did this simple stimuli and complex stimuli, because some aspects of natural stimuli can be decomposed into combination of simple stimuli. And then some others, God knows. We don't even know if the mouse has any memory because they're all living in cages forever for generations, if they recognize an eagle or not. But there are some ideas floating around that they might be able to recognize. So we use something. It's a natural predator. Mice show some looming stimulus response even when they are brought up in cages. Many people have shown that. So
Another potential collaboration.
for sure.
So I don't think there are more questions. Thank you so much for your presentation. It was also very fun to work together. The emails are on the PowerPoint, so please, anyone, please feel free to reach out to the team if you have an interest in this data set. And I know the community is interested in this in these assets. So thank you.
Perfect. Thank you.
Thank you.
So the next presentation is going to be done by Carter Peen, and he's going to introduce to you some of the resources we put together to facilitate data reuse. And so this is going to be including components for each project. So Carter, take it away.
Yeah, hey everybody. As Jerome said, I'm going to be talking about software. So if you found any of those presentations interesting and you want to get into our data, then this part of the talk is for you. So let's see. Okay, so as Jerome talked about, we're using a brain observatory model for OpenScope, and we want, basically we want wide access to the data in our brain observatory. We spent 2 or 3 years collecting high quality data, and in order to maximize the impact, we want it to be reproducible. And if any of you are out there and you've attempted to use open source data before, it can be kind of challenging. So as part of OpenScope's project, we also really want to provide a lot of software tools for reproducible analysis and for accessing the data. So like Jerome mentioned earlier, our data is on DANDI now. There's hundreds of data sets, I think actually thousands now. And that's where all our data is published as of now. VIPA will be published soon, but sequence learning and barcoding are both currently accessible there. Let's see. Oh yeah, DANDI, one note is DANDI comes with a DANDI hub. So if you don't want to have to download the data off of DANDI yourself and you just want to access it in the cloud, DANDI provides this basically cloud computing resource where you can access and analyze their data there. Before I talk about the OpenScope Databook, which will be the bulk of my talk, I wanted to briefly mention that we have also devised the OpenScope Data Portal. If you're interested, you can look at this QR code here. But basically, it's just a web platform that shows the status of each of our experimental session files. So for each of the projects presented today, there's pages there. You can go there. It explains a summary of the project, and it shows you a bunch of metadata about each of the files. So here you can see we have the mouse ID, the session ID, the date. In this case, it shows the probes that we used, and a number of other pieces of information. So I'd encourage you to check that out just to get an overview of what data we actually have available. Okay, so what I primarily worked on for the past 2.5 years is the OpenScope Databook, which is basically our vehicle by which we want to help encourage reproducible analysis. We have DANDI, which basically makes the data widely accessible, but it's not super useful unless you know how to analyze it. We're leveraging the NWB format, which is what I believe everything on DANDI uses. And so the Databook pretty much shows you how to work with NWB files in general, but it also shows you how to specifically analyze Allen Institute's data sets. So any that are released this year or any released previous years, the Databook is a super valuable resource for analyzing those. As you know, I guess for background, already mentioned, we have it on the data available on DANDI, it's NWB. And we don't know of any other centralized repository for super reusable analysis code on NWB data. And there's not a lot just for neuroscience data in general. So we wanted to provide this one centralized location where you can get all of that analysis done. So that's a nice segue into the goals. We wanted it to be really, really simple. We know reproducible analysis is a huge challenge, even after it all comes from the same input format. And we wanted it to be minimal amounts, of button pushes. So ideally, you could take any analysis script, just push 1 or 2 buttons and reproduce all the meaningful figures from a project, or at least the significant ones. We also want to educate users on this, like the actual structure of the data. So NWB files are explained in detail in the Databook, but also the meaning of the data, right? Like the actual context behind what these measurements are referring to. And then create a platform for easily sharing and generating the figures. So ultimately just sort of lowering the barrier of entry for anybody who actually wants to use and collaborate with OpenScope or leverage our data for their own findings. So how do we do this? Well, the Databook is a web platform. You could find it on Google if you Google OpenScope Databook. It's also on GitHub, so you can find it there. And we'll also post links to that as well. Also, if you go to that data portal that I mentioned earlier, there should be links to the Databook there. It's a Jupyter book, which is basically just an application for creating like a front end for Jupyter notebooks. So we have all these Jupyter notebooks that were written by me and other people at the Allen Institute and our collaborators who have presented today. And the Databook just creates a nice, sleek interface for people to actually look at the notebooks and use them. We have them organized into chapters. It's also, like I said, hosted on GitHub, and it's launchable through GitHub pages. And importantly, it also is not just a static web page where you can view the notebook, but it provides a launchable interface to go to various computational platforms. So that includes DANDI Hub, which I mentioned earlier. It includes Google Colab and Binder. You can also run them embedded within the web page, but that's a little bit clunkier, but you can also do that. Or if you like the classic way, you can just clone from GitHub and run the environment there. So there's various options, and hopefully they all serve to make the actual computation super easy. So you're not having to figure out exactly how to reproduce the environment. And I'll talk a little bit more about that in a bit. So as far as content goes, this is not an exhaustive list, but on the right here, you can see this is the exhaustive table of contents. I want to draw your attention to this chapter here, where we have published the latest 3 OpenScope data sets, which is sequence learning, temporal barcoding, and vision to hippocampus. So those notebooks will be particularly of interest to you if you are interested in any of the data presented today. And I'm going to go over those notebooks in more detail in a little bit, just explaining the content. Yeah, so there's all kinds of analyses on the Databook. A lot of the basic analyses you could imagine wanting to be done, like receptive fields for those experimental sessions that have opto tagging. It includes those, all kinds of stuff. And then we also have higher order analyses, like using CBRA from the Mackenzie Mathis Lab, or TCA, tensor composition analysis. And we are still adding content to the Databook. We are also accepting contributions from other people, right? We want to, as part of open science, collaborate. So if there's additional analyses you think belong here, you can either work to add them yourself, or you can reach out to us, and we're always updating this platform. So like I mentioned about computation environments, you can launch to these various platforms. And we did our best to make the environment setup very simple. I think everyone who works with software is familiar with the big challenges with reproducing environments and getting things working smoothly. So this is our current working solution. And in the future, we will be also adding Docker as an option to reproduce the environment. So we're just trying to come up with many ways that that you can pick your poison and figure out how to reproduce the environment most easily for you. And accessing the data. So we talked about DANDI a couple times now. Let me just show you really quick, if you are interested, how you actually can get the data. So you log into DANDI, and if you have the DANDI set ID, or let's search. Let's see. If I search barcoding, you'll see this public DANDI set temporal barcoding, and you can go here. It includes a ton of information about the experiment and the project. And you can actually view the files. So these are organized by subject ID. And you can see the various files associated with that subject. So these are all from one experimental session, and they're different probes. Importantly, if you want to get the path, you'll need this for downloading the file through the API. You press the I, and you'll see the path here. All right, so this is useful. I'll show you why in just a few moments. But that's basically an overview of how to get the data on DANDI. You can also just download them through the UI this way. And if the DANDI set is embargoed, you can use a DANDI API key. So at the moment, the Vision to Hippocampus data set is embargoed, but soon will also be publicly available. Another thing I want to mention is that you don't have to download the data to use it. So in addition to being able to just run it on the cloud, like in DANDI Hub, you also can stream the data. So I'll explain how that can be done in a bit as well. So probably the best way to explain how the Databook works, if it's not clear yet, is just to demonstrate it. So I will get it pulled up here. Oh, there we go. Let me refresh this. All right, so this is the OpenScope Databook. You can see the front page. We have sort of explaining how to use it, more describing what it is in more detail. We have our pretty large, extensive author list at this point, where you can see various authors and how they contributed. Up here, if you click on this, it will take you to the GitHub repository where we have releases. We also have a DOI. So the Databook is published, and it has a DOI that you can find here on Zenodo. So if it needs to be, it's also citable. Oops. All right. All right. So I'm just going to explain a handful of these notebooks very briefly, and then I will explain the 3 notebooks that pertain to the data sets released here today. So if you haven't used NWB files, this is a super useful notebook for you. It also explains how to download them off of DANDI. Let's see. This, to download just a single file, you'd input that path I mentioned earlier here. And then this code will download the file for you. If you don't want to download it and you want to stream it like I mentioned, you could do the same thing with this code. And it will allow you to access the data with streaming. You can get a good view of what the data actually look like here. So NWB as a package has a really useful I guess, output tool, which allows you to just view the NWB file and its contents. And you can see it includes different modalities. It includes the actual electroprobe metadata. It includes our eye tracking data and our stimulus data, subject information, and these are the actual stimulus tables that show what stimulus was shown at what time. So it has all this information. It's really easy to view and access. Okay, so if you're more interested in these, I encourage you to look at these. But now I'm going to go over the just the data sets released today. So to start, I will go over sequence learning. There's an abstract showing here. It shows the sort of the stimulus, the structure of the stimulus shown here in the experimental design. We have an environmental setup step, which I mentioned earlier. It sort of just sets up the environment for you, so you don't really have to do any additional work to get this running. And then we have some cells that actually give an overview of the data. These were generated from another notebook that we have, and it actually just shows here all of the different experimental sessions and information. So this is pretty similar to the data portal, but this was generated actually from the data on DANDI rather than generated from our internal system. And you can extract that into a data frame that just gives you an overview of the subjects. And I pulled out some information. There's 1,197 files. We have 13 subjects. And it gives you an overview of the genotype, things like that. So you looked at this, let's say, and you picked which mouse and file you want to look at. You copied it from DANDI, and you can download them here. We used 2 major stimulus sort of timelines or schemes here, the sequential stimulus and then the random stimulus. So I downloaded one of each to compare. And they're red here. So here is, like I showed earlier, this is a sequential stimulus file with all the information. This is a bit different from an ECFIS file. So because it's 2 photon, we have some other metadata here, which shows the imaging planes. And it includes things like the corrected fluorescence, neuropil trace, also images. And you'll see in more detail in a second. So this is printing the metadata, right? So you can get the actual depth that this particular file was taken out of the subject ID, the plane. You can also see the actual movies, like the videos that we showed the mouse as stimulus. So this one in particular just has 3 movie clips and then also gray. If I were to run this, you could actually watch them and I could play and you could see them in real time. Let's see. Okay, so if you want to do some like preliminary analysis here, you're going to want to select which stimulus times you find interesting to your question. So you can go here, you can look at the stimulus table, and some of this information is extraneous, but some of it you might want to know. And these stim tables are explained also in other notebooks. But basically, this is movie clip B, and I just selected when frame 0 was shown, right? So the movies are shown on a loop, essentially, and I'm just interested in at the beginning of the movie what type of activity happens in response. I do the same thing for the other NWB file. After selecting the stim times, we also want to select the fluorescence, right? We're actually going to extract the fluorescence from the file so we can actually view how it's responding. And you can see here, this is the image taken from the brain. I think this is a max projection image. And we have the segmented cells from this plane. So there's quite a few regions of interest. And we have, let's see, we have our DFF, which basically takes the shape for each cell, and for each timestamp, there's a fluorescence measurement. You can align it. Oh, this is just for the entirety of the session. So this is basically all of the DFF data recorded. I also want to note we have the running information. So like I showed, we have the eye tracking, we have running. So those are our main behavioral metrics here. You can see that. And to get a better idea of how the session is structured over time, I have this code. Just skip past this for a second. Okay. I have this code which shows actually how these two files look in terms of a timeline here. So let me zoom in. This was our random NWB. You can see there's all the different movie presentations shown throughout the duration of the session, and they're just organized randomly. And then just also get an overview of how the data look, the recordings look over time. You can see we have the DFF and the running speed plotted throughout the session. So this is the same plot, but zoomed in to just a smaller interval, just to get maybe a better, clearer view of how the movies are shown and what the DFF and running look like. And then I've done the same thing for the sequential file. So there's a portion in the sequential file which is random, but the majority of it is actually each of the different movies shown in a repeating order. Also with the DFF and the running. Okay, so we've basically taken all the important information from the file. We've taken the stimulus, we've taken the DFF, and we want to actually just view the responses now that we know what everything looks like. And that's what this is doing. It's just getting responsive windows within a 5-second interval. So it's 2 seconds prior to the stimulus time and 3 seconds after. And this is what we see. So this notebook is super easy to run. You can just get these basic responses really quick. And if you want to look at like a different stimulus or a different sort of time window or a sub-selection of cells, you can basically rerun the analysis very easily. And you can see here some cells. So the y-axis here is trials, the x-axis is time. And you can see some cells are very responsive and some cells don't show good responses. And you could expand on this. You could get like z-scores, or you could normalize them in other ways, and just get different views of that. And then finally, I want to point out that we also in several of the files include the raw movie. So prior to extracting the fluorescence, we also just actually have the movie in the file, but it's very large. So it takes a long time to download. So if you are going to look at the raw movie, I'd recommend doing it on one of those cloud platforms I mentioned earlier. And And if I were to run this, you would also be able to just actually watch the movie and see the fluorescence over time. So you could use that for your own analysis, or if you wanted to extract fluorescence using your own code, you can do the same thing. So this is just a quick overview of what the sequence learning data look like. Notebook can serve as a good jumping off point to dig into the data. All right, so now I'm going to go over notebooks, analogous notebooks for the two data sets presented today. Let's see. So we picked the file from DANDI, like I showed earlier. We can download it. One detail about the, I think this was already described in Pamela's presentation, but yeah, so basically we have these different probes targeting different regions. And you can see here, this is just, I generated this from a different notebook as well, which basically shows it has the coordinate information in the file already. So it gives you a really nice idea of exactly where the probes were, and you can see which probes are targeting which regions. You'll note that there's some warping on the probes. That is not that's not from the actual probes. The probes didn't warp during the experiment. They are from mapping the location of the probes onto the model brain during the CCF, the Common Coordinate Framework registration. So yeah, just be aware of that. And now, so we have this code here to get, like I just mentioned, we have the CCF information. So for each unit, we can actually retrieve the location, the brain region that that unit was in. And that's what this code is doing here. And there, you can also select units based on their quality or a variety of other metrics. That is described, let's see, here in this notebook. So units have a number of different metrics that we use to rate their quality, and it's recommended to filter them down based on, I guess, your preference for the threshold for those metrics. In this case, we're just using the basically noise quality attribute and the number of ISI violations. So to filter units based on those, you get 3,000 approximately good units. And you can see the brain regions that they're located in with these acronyms here. Next, we're also going to need the stimulus times here. So we can see these are the different blocks shown here. And in this case, we're just showing the full field flashes that Pamela described that are repeated. It was repeated 90 times. And you can do basically this, which is just essentially generating the barcodes, right? Aligning the responses in time across trials. And what you see for these three example neurons are these really good looking barcodes, or at least two of them. Yeah, so this code is super useful, super easy to reproduce. And the great thing is you could just take it and take any of these files in the data set and any neurons, and in a very short order, you'll have potentially a barcode for them. To expand on that, we see here the PSDHs, sort of just another view of these barcodes that Pamela mentioned. And then this is more information, more details about exactly how to evaluate the barcodes. And you can see they're doing some statistics. This one is just counting the spikes. So sort of how to determine whether or not something is a barcode using different metrics. And it's described in more detail if you want to read it. And then finally, like Pamela talked about, we also want to be able to compare barcodes, right, and calculate the distance between them. So this is using, what was it, the Victor Purpura metric. We're calculating the distance between spikes, or excuse me, between barcodes. And a lot more detail here. And you can see you get this nice distribution of the spike distance metrics. I would encourage you to read these. These also describe more detail about things you could look into the data. And as we already mentioned, I would encourage you to reach out to Pamela to discuss this more if you'd like to expand on it. Finally, I showed this in the sequence learning notebook. We have similarly a session timeline for this data set, which does the exact same thing, but the structure looks pretty different. So we have the unique full field flashes, the repeated full field flashes, the static block, receptive field, and then finally we have optogenetic stimulation. And one more details, we have the receptive fields shown here at the end. So you could basically select which brain region you're interested in, and you could filter them down. And what you get is these nice looking receptive fields for those selected units. Yeah, so that is the barcoding notebook. Many thanks to Pamela and Siddharth for making this. And hopefully you'll find it useful to dig more into their data. Now, finally, I want to go over the Vision to Hippocampus data set, which we just heard about from Mayank and Chinmay and Siddharth. We don't need to go over this again. Let's see. We have same thing. And you can download their data. We also have the probe tracks here. One thing that's not shown in the barcoding notebook is this electrodes table here. So this is the table that actually includes the information of each electrode on the probe. So it shows you basically which probe it's on and the precise coordinates. And from there, you can also get the brain region. So that is what the code in that first notebook is doing. This does quite the same thing. It just selects units based on their quality and their brain region. We have our session timeline here. It's a little bit more varied, and we have many different videos. These can also be, well, I think some of them, the movies can be viewed as stim templates. So you can watch those. We extract the stimulus times in the same way. So this also could be used the same way if you want to just dive into their data. We have a narrower time scale here from 250 milliseconds prior to the stimulus to 500 milliseconds after. And we have code that gets the same thing. Although these are not quite as clear, we'd have to zoom in or change the Vmax in the depiction here to get a better view of the data. But this is essentially doing the same thing, where it's just selecting, getting aligned responses to all the selected units. But we could subselect the responsive cells out of here. Once again, the Vmax should be changed to view it better, but if you look closely, you can see that these units do show some responsiveness. And finally, we also have the receptive fields shown for this experiment, although they're not quite clear in this brain region, but the great thing about the notebook is you could very easily change. Let's see. You can very easily change which ones you're selecting. Oh yeah, back up top, and you could get the receptive fields for a different brain region. So just to summarize and reiterate, I would highly encourage looking at the Databook. We also have these lovely experimental project notebooks for previous OpenScope data sets that were released last year. And any one of these, if you're just trying to get into the NWB ecosystem or just try to learn what the data mean in general, we have all these great notebooks that explain these different analytical concepts and allow you to reproduce them. So to conclude this, just go over a few more things. What are the key challenges with the OpenScope Databook? Let's see. The biggest one is the environments. Like I mentioned, there's all kinds of environment problems that can show up, but we've done our absolute best to make that, obfuscate that problem and eliminate it. And so hopefully the Databook handles that in those different computational platforms that I mentioned. And we're continuing to update the Databook, like I already mentioned also. So we review these notebooks using GitHub. We have that authors list I showed that gets updated automatically if someone contributes. And we want this to continue to evolve and help accommodate the growing open source ecosystem. And this is not a project done by one person. OpenScope has had many people internal to the Allen Institute and external. So this is just some of the people that we want to thank for their contributions, in addition to everybody who presented today. Yeah, that's all I have.
Thanks, Carter. This is fantastic. Oh, we have a echo. I have a quick question. If someone, well, the Databook has gone through some review, and so I thought maybe it was worth highlighting the part that we're reviewing and how this was done. Can you comment on that?
Which part are you referring to?
The entire Databook.
Yeah, so we basically have had many people go through and review the data book. We also have had we reached out to a bunch of different people. Let's see, if I recall, it's Alex Williams and who else?
A couple there on your slides.
Yeah. We've had many people. We've had Yaroslav Halchenko, we've had Alex Williams, some internal people like Josh Siegel, Mackenzie Mathis, and we want to continue. So it's not just a one-time review, right? We want to continue to have people integrate their code into it, and we undergo this rigorous review process with GitHub. And so we think it's really essential for open science to have highly collaborative and utilize these modern tools, right? Instead of just keeping GitHub at this inherently informal place where people just dump their code, right? It's essential that as we are creating these open data sets, that we have, rigorously reproducible code also for data sets.
Thanks, Carter.
Yeah.
This is fantastic. I'm gonna close with a few remarks. So I'll share my. Oh, there might be a question.
Yes, yeah, there's one question in the chat room.
The question is, what is the typical RAM on the DANDI Hub session? Are there ways to set up nodes to run analysis that might take more time to run?
So when you start up on DANDI Hub and you make your account, you log in with your GitHub, you basically can select what type of machine you want to run it on. I think it goes up to 64 gigs of RAM, but if you select that one, it might take longer for your instance to start. So there's also, I think, TPUs and GPUs you can select from too. That might be faster for different types of analysis. I don't know of a way to set up a node to run, but I think the instances take a very long time to time out. I'm not exactly sure on how long, but I think you could run some pretty extensive analysis there.
That's right. Okay, I think we have answered all the questions. Thank you, Carter. Okay, so I'm going to share my screen. Can you see it? As you saw, we put a lot of intent in doing this data sharing. And this is because we're really sort of trying to establish, we think, a complementary models on how we make knowledge discovery in neuroscience. And the OpenScope first round is getting towards its end. And so now we're thinking into the future. And one goal that we had when we started the project was to not only enable team to propose project to the platform, but also to facilitate team to collaborate with one another through the process of data sharing, but also potentially to facilitate team to work together to design an experiment that would help move the field forward. And last year, the steering committee, in the last 2 years, the steering committee was really pushing for this approach. And so they encourage us to innovate in that directions and try out different models. And so we try multiple times. In the first round, it actually didn't work because we didn't have the ingredients. But over the last year, we think we found the process to establish a community project. And to that end, we chose predictive processing as a rallying type of question for several reasons. The first is we found that many of the projects that we have done in the past had some share linkage around predicting the future. And so we sort of decided, okay, so there's maybe a way here to consolidate our community and design an experiment that will serve multiple needs. And so what we did over the last year is reach out on social media around essential questions on predictive processing and collectively work towards reviewing the literature and design an experiment together. This has been a lot of work, but I think we've been successful in that process. So if you go to this QR code, you can go to a document that has been co-written by more than 50 scientists distributed across the world over the span of 10 months. And this was started in April last year. And so we had weekly meetings every Monday on Zoom, as well as collaborative writing into a shared Google Doc that was completely open access. In August, we had organized a workshop at MIT in partnership with the Cognitive Neuro Conference, and discussed potential experiments that would help move the field forward. So on the right here, you see how this collaborative process went. It was a lot of work. So on total, today, there is more than 1,900 comments that have been written by these collaborators across the community to resolve and debate where predictive processing is in the cortex and what type of experiment would help move the field forward. So this document that you can go to, and one really interesting outcome is that what we saw is that some of the OpenScope project you saw today, as well as some previous project that we conducted in the past, seems to have a shared thread. And the thread is related to how we predict the future. Through the review, we identify what we think are 6 principal computational primitives that might be built by the cortex to do those predictions. Those are dendritic computation, which we had a project on, stimulus adaptation, which we had a project with Andrew Bastos and Alex Meyer and Jacob Westerberg, cell type specific computations, E to E recurrence, E and I balance. There are many models that are testing this contribution, as well as hierarchical processing. And so what we found through a review of, and it's a document that's more than 100-page long at this point, but is going to be a review in the next few months. We found is that this computational primitive has been studied almost in isolation by individual labs, but rarely together. And so what we eventually converge onto with that collaborative effort is a shared experimental design that is intended to bridge insight that has been built in the literature. We found that there were publications that disagree with one another, probably because the experimental design was so different that the context of the predictions did not trigger the same mechanisms. And so this experimental design that we are building at the moment is combining multiple experiments that were tested in the community and published upon. And what we want to do is record a set of mice with both 2-photon and NeuroPixel recordings across these 4 different designs chronically to be able to adjudicate if they are the same mechanisms for predictive processing or they are different. And what we hope to achieve with that process is facilitate a common language across the community that is interested in predictive processing and help people consolidate around the shared data assets. So that process of designing an experiment and reviewing the literature is very open. We shared a Google Doc online through the entire world. Anybody can come and make comments and contribute to the piece. The experimental design was also a crowdsourced, and we will, in 2025, collect this experimental design once it's completely finalized and share the data as it goes. So there will be no embargo because it's very collaborative with teams that are distributed. So there's no need for that. And anybody that watched this seminar will be able to access this data set and contribute to the analysis. In the document that I linked to, there's also an analysis plan that is currently being laid out. And so if you're interested in contributing to this analysis, I encourage you to go there and start commenting and interacting with the people that are part of this effort. We meet every Monday at 9 a.m. Pacific time on Zoom. There's a link in the Google Doc, but you can also interact through Slack or through comments in the Google Doc. So this is why I have the slides. So I hope we've shown you today that we are on a mission to sort of enable science in the open, facilitate data sharing, and push our scientific interest forward as a community. And so it's important to me that we built this in a very inclusive way. And so whenever you have interest on some of the data set that we shared today, please reach out to the team, shoot them an email, and then try to sort of just build project together, and so that everybody win in the end. Thank you everyone for your attention.
Thanks, Jared.
Thank you. Bye-bye.
Thank you, everybody. Thank you so much.